Well, I do have mostly a voice today, which is good because the backup sub I got for today needed a backup sub, which is me. So we'll continue talking about DNA topology. I will be traveling on Friday and at a conference on Monday, so those lectures will be pre-recorded, but I will still be available during class to answer any questions. So if you have questions during those lectures, let me know anyway, because I'll still be able to answer questions even if the lecture is pre-recorded. Any questions about anything? Okay. My apologies for frequent coughing, but we will continue talking about DNA topology today. So there are several different forms of topological structures of DNA. One of them, is my favorite, DNA knot, so any closed circle tied in a nice knot. You can also have links. Biologists use the word catenate instead of link, or decatenate to unlink. And so a link to biologists is a catenane. In each of these cases, what we're referring to is, this is actually double-stranded DNA right here. So you've got double-stranded DNA, which you can see in this zoomed-in picture. So this is on the very top, that is unknotted. And you can see if we zoom in that it is double helical DNA twisting around the center line right here. Any questions so far? Recall that linear DNA can circularize if it's long enough and has sticky ends, and then a protein called ligase can then come along and seal these nicks, creating closed circular DNA where the backbone is now fully sealed. A fully sealed backbone does have important topological implications. If I go back and take a look at cutting, so if I go back and cut and create a nick, well, now this is no longer a closed circle, and so calculating the linking number between this and this doesn't make sense anymore, because one of them is a linear strand. To do linking number, you should have a closed circle. And so thus when we talk about linking number, we will ligate it and have, closed circular DNA. So the phosphate backbone is completely connected, and so our double-stranded helical, DNA, we can talk about the linking number between one of the two backbones with the center line or with the other backbone, either way, and then this represents nice unknotted DNA. Remember that we knew that linking number equals a twist plus writhe. In this example here, this DNA has 360 base pairs. In most in vitro experiments, so in the test tube experiments, the DNA is usually about 4,000 to 7,000 or so base pairs, but both longer and shorter are sometimes used as well. And a mini circle like this actually has very important applications. In this particular case, you can go ahead and count that as we go long, red crosses over once, twice, up to 5 times. And so if I calculate the linking number going all the way around, I'll get that the linking number link equals to 36. In this example, our twist also equals to 36, and our writhe equals to 0. Since this thing does lie in a plane, then all projections except for the one that goes vertical to here, that would just project it to here. All projections that are not from the plane, every other projection will have 0 crossings, and thus we know our writhe is 0. Link equals twist plus writhe, so twist is 36. And so we have what's called a relaxed DNA molecule. So when twist is as happy as it wants to be, so DNA does like to have generally about 10.4, 10.5 base pairs per turn, but that does depend upon salt conditions. For simplicity, we are going to assume that the salt conditions are about 10 base pairs per turn, and so thus since we have 360 base pairs, if we divide through by 10, 360 divided by 10 gives us a twist of 36. And so we'll assume salt conditions of about 10 base pairs per turn, but usually it's a little bit higher than that. Integers are just so nice to divide through by, though. So when twist is, where it wants to be, so the preferred twist, whatever the salt conditions happen to be, and thus writhe is as small as possible, that's when we say that it's relaxed. So writhe close to 0. I say close to 0 because remember, these two things do not need to be integers. So while link must be an integer, neither twist nor writhe needs to be an integer. So you can imagine if you are working with a belt or with a cord, and we put just a little bit of, just make it a little bit out of the plane. Also, DNA likes to have about 10.4. If we've got 360 base pairs and we divide through by 10.4, we're no longer going to get an integer. And so when things close up, they'd like to close up exactly like this, but sometimes the preferred twist might bring it in maybe at a 90-degree angle, maybe at a 34-degree angle. But to close up the backbone, you have to have them matching perfectly. So we need to have this perfect matching, and so thus these two, twist and writhe, may not be integers. So maybe your relaxed form might have a writhe of 0.5 or minus 0.4, but it's called relaxed if it's close to 0. Suppose we now take a protein that cuts the DNA and changes the linking number. So basically what we have is going from, let me actually clear my screen so I have a little bit more room. We might be going from something that is all, has this sort of linking number. We can then cut. So we'll cut this, allow the other strand to pass through, and so now, we can now remove these crossings and now get something that looks like this. And so we can change the linking number via a protein, a type of topoisomerase. In this case, the topoisomerase that was used for illustration was called gyrase. There are lots of different topoisomerases. So now we have a linking number of 32, so we removed some of the links. But DNA still likes to have a preferred twist. So since it likes to have a preferred twist, what happens is the writhe changes. So the twist stays the same. So here we have our link. Our link equals to 32 now. Our twist likes to stay the same at 36, which means our writhe must be minus 4. 36 minus 4 is 32. And so thus we now have supercoiled DNA. Now, in reality, if I change my linking number by 4, that 4 will actually be partitioned between twist and writhe. So in reality, your twist is not going to remain at 36. It wants to remain at 36 because that was its preferred twist. But that change of 4, maybe your twist will now be 4.9, and your writhe will be minus 2.9, so that we now have a linking number of 32. So twist will actually change in reality. Maybe about 70% goes into writhe and 30% goes into twist. It will depend. So, but this is basically for illustrative purposes, assuming everything, goes into Rive, then the Rive will change by -4. But twist does change a little bit as well. It would prefer to stay at 36, but there is strain between the twist and Rive, and so they sort of compete with each other. Most of it generally going into Rive. Any questions about that? Notice that this DNA is negatively supercoiled. So that's actually important, that it's negatively supercoiled, that the Rive is a -4 or a negative number. And the reason for that is DNA is usually negative supercoiled. If you want to replicate DNA or transcribe DNA into RNA, then you're going to have to pull the two strands apart. And if the DNA is underwound, because this thing is negatively supercoiled, that means your DNA is underwound. And if it's underwound, it'll be a lot easier to pull the two strands apart. In fact, they might open up naturally in certain spots, allowing your replication machinery to come in, or maybe proteins might come in and help to open it up. But in either case, you can open it up and then have some proteins coming in to do your replication or your transcription. Note that in this case, we still have the same link equals to 32. The twist now, however, is 32, and the Rive is 0. In this region, the twist density is identical to this. If I zoom in here or I zoom in here, I see identical pictures. These two pictures are identical. But by opening this up, we can now have that 0 plus our 32 allows us to have the same density in this region here. Versus if I didn't have it underwound, if I had to open up this, well, then it would have to supercoil, because now I'd be forcing more twist into this region. So if you try to open up this, that'll strain this part here and cause it to supercoil, because it would now need to, overrun to make up for that. So questions about that. So supercoiling has very important biological implications. DNA is generally negatively supercoiled. And here you can see pictures of negatively supercoiled DNA, different densities, as well as relaxed DNA. So sometimes you'll see the word substrate. Depending upon the experiment, it means the starting conformation. And in today's talk, all our experiments are on DNA, so it's the starting configuration of DNA before it acts. And usually the starting conformation is unknotted and supercoiled, but in some cases it is relaxed or nicked as well. So normally supercoiled is your standard substrate, but we'll actually mention a few experiments where the DNA was actually nicked. And if it's nicked, well, if it's nicked DNA, then it can have whatever twist it wants. So this could be either relaxed or it could be nicked. I can't tell from the picture. From the paper, they said relaxed, but it could have been nicked as well. They would look very similar. Any questions about that? Your DNA is actually linear, but it's tacked down to a scaffold. So this again represents double-stranded DNA. So we've got double-stranded DNA here, which then supercoils as it comes out. When your DNA needs to be accessed, it's generally bound by a whole bunch of proteins. So it's generally wrapping around proteins. So it's generally wrapping around proteins to keep it nice and organized. But if you need to transcribe a gene, then the portion of the DNA containing that gene must come unbound for the proteins, and you might see this sort of thing in a supercoiled type fashion, where this form is called plectonymic. Let me rewrite that. Plectonymic supercoiling. And that's what you see here, nice plectonymic supercoiling. You can also have solenoidal supercoiling, which generally happens when it's wrapped around the protein. So in this case, we're wrapping around a nucleosome, wrapping around about 2.5 times, and you can see it's crossing itself, and so we've got writhe here. So if I try drawing that again, it's going to be, we have some writhe here where these things are crossing itself. And this type of wrapping is called solenoidal supercoiling. So you can either have solenoidal supercoiling, which is basically like this, wrapping around proteins. Maybe it wraps multiple times before it goes to the next one. And then you also have the plectonymic, where it wraps like that instead. So this would be the plectonymic supercoiling. Questions on that? And here we can see the nucleosomes compactifying more and more. There's actually some questions as to exactly what the shape of DNA is in your body. They can take pictures and see what the chromosome looks like. They know about this nucleosome wrapping 2.5 times around, what happens in between, compressing, but your DNA is highly, highly compressed in your body. It has to fit a lot of material into a tiny cell and kept very organized. You can imagine why your DNA should be unknotted and nicely organized with the proper supercoiling. So I packed for a trip. I had to put in my extension cord into my bag. Is it easier to pack it if it's knotted or unknotted? It's a lot easier to pack it if it is unknotted, and then I will likely wrap around forming this solenoidal supercoiling in order to pack the DNA in more easily, or my extension cord. Working with an extension cord that has knots in it, it's problematic. You can't work with it as easily. And it's the same thing for DNA. Keeping it unknotted and very nicely organized just makes it much easier to work with. There are a number of computer simulations. So this is a computer simulation of nicked DNA. A colleague, John Simon, because you have in vivo experiments, that's actually in the cell. So those are the most relevant ones, but the hardest to do because there's so much going on in the cell. In vitro experiments, which is in your test tube. But we can also do in silico experiments, and that is computer simulation. I'm hoping to change the word in silico to one that would make much more sense, and that would be in virtuo, okay, I can't spell. The first time I heard in virtuo was from John Simon back in the 1990s, and maybe this one will start taking over as well. I think people say both of these, but I like in vivo, in vitro, in virtuo. Makes sense for your computer simulations. So when I was new to this field, sometimes I'd be listening to a talk and wonder, wait a minute, was that a real experiment or was it a computer simulation? Computer simulations are quite good, but it's important to know which is which, and so one should pay attention to whether it's a computer simulation or in the test tube or in the cell. Which it is does give you information as to how best to analyze it. Okay. And then we've got, again, in virtuo, so in the computer simulations of supercoiled DNA here as well. Okay. So remember, when DNA replicates itself, it has to pull things apart. And so we pull things apart. And we can, though, because we've got all this twisting, we get these precatenanes. So this is partially replicated DNA. which after it's done replicated might actually be linked. Remember, they're called pre-catenanes because links to biologists are generally actually called catenanes. So we need proteins that will change crossings in order to unlink the DNA. A topoisomerase II, that will break. So here we've got double-stranded DNA. It breaks both the strands. So both those strands get broken in order to allow another double-stranded DNA piece to pass through. But there are many different types of topoisomerase. In fact, there's many different type 2 topoisomerases, as well as many different type 1 topoisomerases. In type 1 topoisomerase, only one of the backbone strands, so only the red strand here is broken. So only one strand is broken, and then we pass through here. And so that's the difference between your type 1 topoisomerase, one strand of your double-stranded DNA is broken, versus for your type 2, so type 2, both strands of the double-stranded DNA are broken. But there are many different type 2 topoisomerases, as well as many different type 1 topoisomerases. As mentioned last time, they are involved in replication, transcription, unknotting, unlinking, supercoiling, etc., as well as the targets of many anti-cancer, as well as antibacterial drugs. And you can imagine why they are targets, in particular of anti-cancer drugs. You have cancer cells that are rapidly replicating. If you slow down the topoisomerase, well, then when it comes to replication, so we had one molecule gets replicated, but now it might be linked. These two things need to become unlinked so they can go into different cells. But if you slow down the topoisomerase so it can't do this, then the cell dies because you can't separate these things out. It is, by the way, more than just unlinking. You have to unwind this thing here, and there's different ways of unwinding it. You might have a topoisomerase 1 unwinding that actually changes the linking number. You also could have a helicase, which is just pulling the strands apart. So you can just think of wrapping two ribbons together and just pulling it apart, but that tightens the twisting in the two ends. You need a topoisomerase to now release that tension. And so you have both topo 1s and topo 2s acting in these things, both with the supercoiling changing that in order to unlink it. So if you start slowing down these topoisomerases, then you're replicating cancer cells or hair cells or stomach cells. They will have issues, and that's one way you can kill your cancer cells. So generally speaking, they will unknot the DNA, but sometimes they can also knot the DNA too. I mean, these things are just acting locally. So if you took a big rope and tied it together so it was a sealed circle, but you knotted it first, if you then randomly did crossing changes with your eyes closed, can you guarantee you're going to get the unknot? What if you didn't do it randomly, but you still did it with your eyes closed and only looked locally? Could you bias it towards getting the unknot? Topoisomerase actually does bias it towards getting the unknot, depending upon the type of topoisomerase. So one can create a bunch of knots. So this is a nice knot table from knotplot.com, but this one here on the next slide is actually my favorite, and that's of DNA knots. Now this is actually duplex double-stranded DNA knots produced by type 1 topoisomerase. Type 1 topoisomerase can only cut one strand, but the DNA was nicked. So the DNA was nicked, so topoisomerase only had to cut the other strand and allow it to pass through. In fact, in the experiment, the DNA was likely very highly gapped. So in the beginning, they didn't know how to nick the DNA in just a single spot. So there were probably many things missing. And so topoisomerase just had to cut one thing, and then it could create these DNA knots. So we've got knots, 9 crossing knots, a composite knot between this 5 crossing and this 4 crossing knot, lots of 6 crossing knots, 7 crossing knots. It basically recreates the knot table. The substrate does matter. In this case, where the substrate is supercoiled DNA, and we're working with a type 2 topoisomerase, and you can see how the supercoils are trapped by the topoisomerase. So basically, we went from this supercoiled thing, and then these two sites came together, and a topoisomerase, if you change this crossing here, you can go from the unknot to this twist knot right here. So in this case, the substrate, the type of substrate does matter. In this case, we only get knots where we've got this clasp region along with this supercoiled region in this experiment. So biologists have been creating knots for some time. This one was 1991. The other one was possibly in the 70s or 80s. I forget which. But the main thing is it unknots the DNA. So it simplifies the DNA actually below equilibrium value. Well, first off, what is equilibrium value? Equilibrium value is if the topoisomerase were to act randomly, or if you were to take linear DNA, but with sticky ends, and you circularize it. So with the linear DNA with the sticky ends, it will circularize. And most of the time, it will circularize to form the unknot if it is only about 4,000 or 7,000 or 10,000 base pairs. Most of it will be unknotted. If it's very long, then you can start getting knots. At about 7,000 base pairs, you don't get a lot of knots. So most of it is unknotted. Generally speaking, I should be having it supercoiling, or not supercoiling, but it might supercoil around. So it might look like that. And then you'll also get a few knots out as well. So your products, when circularize, you can get knots as well. So you get a small amount of knots and a bunch of unknots with varying amounts of supercoils. That's generally the product if you just let linear DNA of about 7,000 base pairs circularize. You can also have topoisomerase 3, which is a type 1 topoisomerase, to also act on nicked DNA. So the substrate here was nicked DNA. So in this experiment, it was nicked circular, so circular nicked DNA. And type 3 topoisomerase acted on it. And depending upon the concentration of topoisomerase to DNA, you get a certain amount of knotting. So topoisomerase starts to create more knots. And so you do have some knots here. And so on this axis, this is the probability of knotting. And so here we get more knots compared to this right here. And over this is the concentration of topoisomerase to DNA. At reasonable concentrations of topoisomerase to DNA, you get very little knotting. Even if you gave it knotted DNA, you still get very little knotting. It can be as much as 80 times lower. You can also give the, as your substrate, you might also do linked DNA or supercoiled DNA. So they say simplification of DNA topology. This applies to both links, supercoiled DNA, and knotted DNA. Some type 2 topoisomerases, not all, but some type 2 topoisomerases will simplify your DNA well below equilibrium value. So that's kind of interesting because it's, how does it know this is a protein acting locally, making this global change below thermodynamic equilibrium? Now, if you increase the concentration of topoisomerase, then you start getting more knots. And so that was the way they created these DNA knots. You had high topo 2, high T4. There was high concentration of topo 4, and because of that, it created more and more knots. They believed that maybe the topoisomerase was binding on some of the crossings, stabilizing them. And if you have too much topo 2, you start actually getting DNA knots, which you can see in this graph. And these are 3 different types of topo 2, including a human topo 2. We would all be dead if we did not have nice acting topoisomerase. And so these different topo 2s all are not below equilibrium value. Unless you're at a high concentration, then you can start getting DNA knots. Any questions about that? So what product would you expect if topoisomerase acted exactly once on the following 5 crossing knot? Well, we could change this crossing, any of these crossings. And if you start changing those crossings, well, if I change either of these 2 crossings, I get the unknot. So I would get, this picture here, where, I've got these crossings here, but I could pull these apart if I change either of these, and I would go to the unknot. Or I could change any of these 3 crossings. If I change any of these 3 crossings, well then I will lose 2 crossings. Say if I change this, I can lose 2 crossings, and I will preserve having 3 crossings. And so any of these 3 crossings would go and give me this 3 crossing topo knot, which is also drawn here. Or I could draw a more complicated diagram, so I might draw this more complicated diagram of this knot and change a different crossing and get something that might have even higher crossings. So I could get this 5 crossing knot, or a 6 crossing knot, or a 7 crossing knot. You can get higher crossing knots as well. Well, in the lab of Len Ziedrich, they did this experiment where they used a topo 2. Now, the topo 2, I forgot to mention that the topo 2 that are knots below equilibrium value, so let me go back and put that in there, is it uses ATP. All of the ones that are not below equilibrium value does use ATP. In other words, they use energy. ATP can be used as an energy source. And so energy is involved in this reaction. If I just do crossing changes at random without having any sort of energy, you will get thermodynamic equilibrium. You would expect thermodynamic equilibrium. But this energy should be involved in going below thermodynamic equilibrium. And so we've got topo 2, and actually you could use ATP or you could use AMP, PNP, a version similar to ATP, but this thing, so you can only act once, or use a very low concentration. So you'd expect that basically the topo 2 acts one time per DNA molecule. Well, what would you guess if it could act exactly once per molecule? I'd expect, some unknots, some truffle knots, maybe some higher crossing knots, maybe. But they basically only saw the unknot. They didn't see this. Now, I put light gray because in terms of the methods to see things, the methods where they visualize this thing is they run it on a gel, and they saw their product. So they see a 5-crossing product, and they see the unknot, but they didn't see anything else. So it looks like the only thing they got was the unknot, that somehow topoisomerase acting locally knew to change one of these 2 crossings and not any of these other crossings. They didn't see the truffle knot whatsoever. Now, maybe there was some truffle knot, but the method of visualizing how much product you get of different crossing numbers, it's not really known how much concentration you have to have. So maybe there was 20% truffles, but the band was just too light to see it. They did something called gel electrophoresis, where they ran this and the more compact thing traveled faster, and so this 5-crossing product would travel faster, the 5-crossing substrate. So this was our substrate, the starting configuration, and it would travel faster than the product, and the product that they seemed to get out was basically unknots. So big question as to, how can topoisomerase act locally, determine the best reaction to go below thermodynamic equilibrium, and more than that, go straight to the unknot. Now, this is at the moment still an unpublished result. Her graduate student, who is actually a graduate student of both Lynn Ziedrichs and DeWitt Sumner's, she was actually a math graduate student who did this experiment. She did a bunch of other experiments, graduated before doing the control, so this has not been published. The control experiment would have been to have a substrate who can't go this knot right here, this 5-crossing torus knot. There is no way to go straight from this to the unknot, and so they should do the control experiment to make sure the protein didn't act twice. They don't believe it acted twice, so maybe in reality, if we go back to this experiment here, it did go to the truffle knot and then it acted again to go to the unknot. They can't rule that out, but if they did the control experiment under similar circumstances and you see truffle knot, that's good. If you see unknots, well then that means the protein had to have acted twice because the unknotting number, the minimum, the mathematical lower bound for going to the unknot from 5-1 is that the topoisomerase would have had to act twice. Any questions on that? Okay. So there's a big question as to how can topoisomerase act locally determine a global result. There are many thoughts, but one of the main thoughts is this bending. So this is one of the speakers at the December conference will be Alex Vologodsky, and he hypothesized back in 2001 that the topoisomerase might bend the DNA. And if it bends the DNA, then when it comes through, it might more likely want to go this direction. So it might want to bias it towards going here. So if it encounters this, it may not do a crossing change, but if it encounters this, then it does the crossing change. So based upon the geometry, the local geometry, it will determine whether or not to do the crossing change. So in this case, the local geometry, it won't do the crossing change. Plus, this strand is likely to wander off. This strand here is kind of trapped, and so it's not very likely to wander off. Remember, all of these things are moving. Nothing is static in the cell. So if you've got, your topoisomerase and your DNA moving around, once it gets trapped in here, then it might decide to go through, versus if it's wandering around on the other side, it's probably not likely to do the crossing change. So this bent DNA is one of the major thoughts as to how it might be going below thermodynamic equilibrium. And you can see here, in terms of if I change this crossing here, even though I'm only looking locally, I have a feeling that changing this will help organize the DNA, changing this crossing right there, changing that crossing, then I can pull the two things apart, versus would I want to change this crossing? No, that would more likely create complications bringing these two together. So you probably don't want to go from this to this. You probably don't want to do this, but you do probably want to unhook these things here. So you probably do want to unhook. So this is a bad color for this. So you probably do want to unhook, this thing here, do a single crossing change to unhook. And so that's the sort of idea is, if you take a look at curvature, no change, maybe change, oh, definitely change. We now have a nice hooked juxtaposition. So this last one being a hooked juxtaposition. So that would be one way. If you just had a long piece of circularized rope, that if you went around and felt the crossings, if you felt the crossing was hooked, then you could go ahead and do the crossing change. The protein, however, may also be putting in bending. So the protein might actually be coming in here and bending one of the two segments, of the involved in the crossing change. So here you can kind of see that if we've got this, the protein comes along, maybe it was pre-bent, or maybe the protein binds and bends it. And then after it's done the crossing change, it goes off. Here, it's not hooked, so maybe it tries to find something like that, where they're both bent. Maybe it bends it first and waits for another strand to come through. There's still a few questions about that. So here, no strand passage. Here we get topo 2 acting, doing the strand passage. Questions there. One can do this computationally, and there's different ways one can do this. This thing is on a lattice. You don't have to work on a lattice, but there are lattice models as well as off-lattice models. The nice thing about working on a lattice is you are basically, let's take a look at a two-dimensional lattice. In a two-dimensional lattice, you're only allowed to go through points. So if I want to do my DNA, let's do our DNA in red. My DNA must go through the points on my lattice. And so if I want to create a hooked juxtaposition, I can just search for the DNA that goes through in this conformation. So we search through these things here. And then we search for anything where we also have this bent DNA here as well. And we can do hooked, or we can do half hooked, or we can do free. And one can do crossing changes, so we generate a bunch of different DNA molecules and then look for hooked, half-hooked, or free. And in one computer simulation, only do the hooked, only change the crossing, so only go from this to an unhooked sort of thing. So this isn't the one that they did, but I'll just draw it for simplification. They kept the DNA length the same, so they actually, in order to keep the DNA, they did something like this instead to keep it, but then they unhooked it. So they had topoisomerase act only where a hooked juxtaposition was seen. They also did it where you have half-hooked. They also did it with free or just arbitrary. They also did arbitrary, and they compared the two, and sure enough, hooked did get you below thermodynamic equilibrium. So that's one way of getting below thermodynamic equilibrium. And that is it for the topoisomerase part. I still have a little bit on recombinase, not much, but any questions about the topoisomerase part? So main question is, how do you go below thermodynamic equilibrium? What is a hook juxtaposition? And there are different computational approaches. You can do lattice models, and I forgot to mention for the off-lattice model, you might be looking at sort of angles. So maybe you look at the angles between the crossings, and maybe depending upon various different, you might force the bend. So you might put your topoisomerase specifically in a bend. So for the off-lattice, force this bend when you're doing this generating, or look specifically for a bend, and then look for another DNA strand that's close to it. And then if I rotate this thing so that they cross over, maybe I want a particular angle theta here as well that you might be looking at too. And so one can take a look at bends, angles, compare them, do a bunch of computer simulations, do restrictions as to when you do crossing changes, and then see that you also get below thermodynamic equilibrium. Any questions about that? Another protein that can knot and link DNA are recombinases. So recomb means it will recombine, and ACE means it's an enzyme. So like we had our topoisomerase changed the topology, so it was an enzyme. That was the ACE part. ACE means it's an enzyme. Topoisomerase means it changed the topology. The recombinase is an enzyme that recombines the ends. So it will break the two DNA ends and switch the two. So this black head, which was attached to the black tail, well, now the black head gets attached to the white tail, and we've changed the DNA sequence. So that's what recombinases do. They can change their DNA sequence. In this case, our black head joins to the white tail, and so whatever this DNA sequence here is now broken, and we have a new DNA sequence. So the old DNA sequence was all black, now it's half white and half black. And similarly, this white head joined with this black tail, creating a new DNA sequence. And depending upon the orientation, if this head joins to this tail, I create two components. And so I now have two components, which could be linked or unlinked. Or if I join this tail to this head and this tail to that head, then I still have one component, but I may have knotted the DNA. So direct repeats where head to tail, or inverted repeats, head to head, tail to tail, makes a difference into what you can create. And lots of DNA knots and catenanes are made by recombinases. We'll talk more about that next time. We'll start off the class with about 20 minutes on this type of stuff, and then we'll talk more about persistent homology applied to phylogenetic trees. Any questions? So I won't see you Friday, but I may hear from you if you've got questions. Friday's lecture will be pre-recorded.